Welcome to section 2.5. This section is entitled Membrane Proteins, but we're going to talk about kind of a variety of things that are going to be part of the phospholipid bilayer. Because we left off just saying that all membranes have this phospholipid bilayer. You can kind of see here, you have the head regions, the tail regions, hydrophilic, hydrophobic. But there's more stuff that's intermingled. It's not just about phospholipids. So we'll talk about, there's these little yellow guys here that'll be cholesterol. There's these guys here on the surface that are kind of white and red. Those will be carbohydrates. And then these blue guys here will be proteins. So we're going to talk about kind of what each of these does with an emphasis on the proteins, because proteins do a lot of stuff. So we will emphasize that. So for starters, one of the reasons we care so much about this is because we've said that membranes are semi-permeable. That ultimately, especially small nonpolar things can slide through these little gaps between the phospholipids as they bump around. But things that are charged, so if we have ions like K plus or calcium 2 plus or Cl minus, things like ions in certain larger molecules, especially larger and polar molecules, can't slide in. They can't get through that fence, if you will. And so we need to have some other way to get them into our cells because we do need them. And so one of the ways to go about this is going to be proteins. And so we're going to focus on these proteins that are embedded in the membrane, kind of like a door or a gate to the fence, that will allow specific things in. So each of these proteins will act like a gate or a channel. You can kind of think of them as like tubes that perforate the actual membrane. So this would be like our little phospholipids. I won't draw the tails, just assume it's like this. And you'd have this big protein that would be kind of like a straw that we stuck through the membrane that allows stuff to go through the straw, through the protein. But it only allows usually one ion type. So like there'd be a specific protein for just K plus, potassium. There'd be a specific protein for just calcium ions or chloride ions or whatever other molecule we're talking about. I mean, this can work with some molecules as they get bigger, but it won't work with huge molecules. Like you might be able to get a glucose through, but don't expect to get another protein through a protein because it's just too big. It just doesn't work. It's like trying to get a piano through that small gate. Eventually you have to have other means, which we'll cover later. But I want you to make sure you realize proteins will be doing a lot of the transport, and ultimately they're very specific. So you need to have lots of different types of these membrane proteins to allow us to get all the various things we need in and out, especially ions. Now, moving on, there's two big types of proteins. The ones we're going to focus most on is these integral proteins, and specifically from those, the transmembrane ones. And so what that really means is they're saying that these are the proteins that go through, you can see right here, they go through the actual membrane. So that allows something to kind of come from one side, go through the protein, and get clear to the other side. There are other proteins that you can talk about. These ones are called integral proteins, but they're called monotopic, and that just means they stick into the membrane, but not through it. And so they can be used for other stuff, but they ultimately will not be used for transport. So they'll be a little bit more like our other type of protein called peripheral proteins. So peripheral proteins are more like taped on. They stick to this like polar head region, so they tend to be hydrophilic. So they're going to stick to the surface or stick to a protein that's transmembrane or monotopic. They're not actually going to go into the membrane. So it's more like taking duct tape or something and sticking it onto a surface versus a nail is more like these integral proteins where it goes into the surface. And so these guys can still have a useful purpose. They can be used to attach stuff because we have lots of things that make up kind of like a skeleton that we'll get more into next section that help keep the shape of a cell. So they need somewhere to anchor on the cell membrane so they can work as like anchor sites. They can hold other things like enzymes or pigments just because they're a set place where you know where they're at by sticking them to a section of membrane. So there's a bunch of different things that we can ultimately stick here. But what I want you to realize is they're not going to be used for transport. That's like the major thing that I want to make sure I get across. The integral transmembrane guys, those are our transport proteins. These other guys will serve all the other cellular functions that we need, but not transport. All right, now we move on. We can get a little bit past just this idea of proteins. So now we're going to discuss some of the other stuff that's in there briefly. And so we're going to call this whole model of what we refer to the phospholipid bilayer as, when you mix in all this other stuff, we call it the fluid mosaic. So the fluid part is that phospholipid bilayer, where we already said before, they're bumping and moving. They're dynamic. It'd be the word they often use, not worm. And the mosaic part is all the other stuff that we stick into it. because it's not just a phospholipid bilayer. It's much more than that. And so in this case, it's going to have those proteins we talked about, but it's also going to have cholesterol, and it's also going to have ultimately carbohydrates. And the way these will work, let's see if I can play this video, you can see here the phospholipids, how they bump around and shift, how it's very dynamic. You can see there's other stuff attached. There's some carbohydrates you can see sitting here. Cholesterols are wedged in. The cholesterols will help with the shape of the cell. So I get myself clicked here. So they can help with the shape of the cell, and they help keep the membrane fluid. They help keep it flowing. The proteins we've already discussed, so we've already kind of said what they did. And the carbohydrates will kind of stick out on the outer surface. You only typically find them on the outside surface because they'll be used as an ID card of sorts. So as cells come by, like our immune cells, they can read these little carbohydrate chains, these sequence of, usually it's like say 10 or 20 monosaccharides stuck together. But which monosaccharides and how many helps determine whether it's your cell or a foreign cell or a foreign body, whatever you want to consider it. It could be a virus, whatever. So if it doesn't have the right carbohydrates, it sets off the alarm, which then triggers the immune cells and the immune system to get rid of it. So this is very important, even though it's not part of our transport, and it's not so much, I guess, a cellular function as it is a function of a multicellular organism's whole body working together, our immune system. But it is critical to keep us alive, because without this, we wouldn't be able to detect that we're sick, and we wouldn't be able to heal ourselves to make sure we don't stay sick or get sick enough that ultimately we could die. So this is a critical function on the outside of the cell. It's your ID card. This is very important if you need like blood transfusions or especially an organ transplant, because these markers, if you don't have the right ones, can trigger things like rejection, where you attack the blood or attack the organ, and so you'll ultimately not be able to get the benefits. You'll still end up, in the case of an organ donation, you could still die. And there are some kind of cool things we can do with the cell membrane where we kind of extend it a little bit, these extensions. These aren't hair extensions. These are just going to be parts where we take this membrane and we add stuff, typically proteins, that are going to allow us to have these pieces that stick out. And so these protein pieces that stick out oftentimes have a motor that allows them to go back and forth, or in some cases spin, and we're going to use them primarily for motion. Now that doesn't mean it's going to just make the cell swim. You'll see that both cilia, these are the ones here, you'll notice there's a lot of them. They look more hair-like. I like to think of them as more like oars, where there's lots of oars to make the boat move, but each oar is relatively short, versus flagella, which is more like the tail. Like think of an alligator or a crocodile, where they can move the tail back and forth and propel themselves, or a fish. But you don't typically have a lot of those. There are some things that do have 2, 3, 4, that do have multiple flagella. It's not like all ones are kind of like human sperm cells. We think they have one tail, or like a fish or a crocodile that has one tail. There are some that will have more. You can see several here, but they'll be longer, and they're normally used for movement of some sort. Now sometimes it's making the cell swim. Sometimes it'll actually be like pushing water along, so the cell itself is like in place, but as it moves, it draws water across it. And so some filter feeding organisms can do stuff like this, where they beat the flagella, which allows for the water to move past them so they can feed, so they can pick off the little particles, the little bacteria or algae from the water as it passes. We have cilia in our bodies, in our respiratory tract, and so they're actually used to help haul mucus up from our lungs and then dump it into the back of our throat where our esophagus is, which then makes the snot or mucus that you just pulled up essentially go down into your stomach, where any bacteria that were in there should be killed by your stomach acid. So if you ever get sick and you're like, oh, I feel like there's snot at the back of my, it's because there's snot at the back of your throat and you're swallowing it. That's your body getting rid of it from your lungs. If you smoke too much and you clog up those cilia or you damage them, that's why you have to like hack up. And so you'll see sometimes smokers who've been smoking for a while coughing up into like a handkerchief and like pulling up these huge mucus loogies, and that's why is the cilia aren't working, so they have to bring it up in chunks by just hacking at it. So these are two of the guys that we'll have to help us move. They're also going to be part of that membrane, at least they're attached to it. There'll be protein extensions, and that wraps up the membranes for now. I'll see you guys tomorrow.